New cells are made by cell division, and cell division could be split into two categories. One is mitosis, which is normal cell division, which makes sure all the cells have the same number of chromosomes, they are genetically identical, or meiosis, which is the formation of the gametes. The gametes are not genetically identical and they contain half the number of chromosomes as a body cell. Mitosis has many uses. The key one is growth, the formation of new cells. All growth occurs after the two haploid cells, the sperm and the egg, fuse to make a diploid cell, which is genetically identical to all other cells in the body. This is done through the act of the expression of the genes that are specific to that cell, so the cell becomes specialized. Another function of mitosis is the repair of damaged or lost cells. Again, these cells need to be identical to the original cell. Contrary to popular belief, not every single cell within the body can divide by mitosis. Very few cells actually retain the ability to do so. But if they do, they can make genetically identical daughter cells with the same genetic material within. The only time this can change is through mutation. Another thing about genetic material is what we consider the classic cross chromosome is actually a little bit of an error. Chromosomes are actually single-stranded, they're not cross-shaped, and they're called chromatids. It's only when they're duplicating do they become the classic cross that we know about, which is the identical sister chromatids. However, both can be referred to as chromosomes. So the single strand there on the left and the double stranded ones on the right, both known as chromosomes, but to be really accurate, you've got a chromatid and then identical sister chromatids. From GCSE, we should know that the parent cell has the chromosomes within them, and then they replicate the genetic material, becoming the classic cross as we know them. They then line up in the center. The genetic material then splits and starts to divide, and we end up with two cells forming with four chromosomes in. Four at the start, four at the end. They are genetically identical. However, we need to know the different stages. They are interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. We don't worry about prometaphase and other subdivisions within there at this level, just the standard stages. The first stage is interphase, which is actually split into three distinct areas, G1, S, G2. Interphase is often referred to as the resting phase of the cell, when no division is taking place. But there's a lot going on. This phase is when the DNA is actually replicated, and it still stays in the form of chromatin, so it's not visible. The nucleus appears cloudy during this stage. There are, however, three distinct phases. The first is G1, where the proteins are produced. Basically, the cell is making more proteins to manufacture organelles. It then moves into S, which is the synthesis phase where DNA is replicated, and interphase finishes with the second stage of growth where the organelles are actually made and energy is stored up for the act of mitosis. There are a few other features of interphase we need to know about. The chromatin is not visible. You can't see it. The nucleus appears cloudy. However, during this time, the DNA, the histones, and centrioles, those two little structures at the bottom of the picture there, are being replicated. You've also got replication of all the cell's organelles, such as the mitochondria and stuff. That's happening in cytoplasm. Of interest are those histones. Histones are proteins that DNA is coiled around, a little bit like cotton around a cotton reel. It allows the DNA to condense down and become visible as the chromosomes. So normally it's dispersed throughout the entire nucleus, but as replication occurs, the genetic material is wrapped around the histones and we end up with it condensing down and becoming the chromosomes, which you recognize as the cross. The next stage is prophase. Prophase is recognizable because the chromosomes become visible as the classic cross shape we recognize. Also, the centrioles have moved to the poles of the cell so they can hook the chromosomes and pull them to each end of it. So the chromosomes become visible. It prevents them tangling up with one another because chromatin is way too dispersed to be able to maneuver around easily. They also replicated fully, so they end up with two identical sister chromatids, and they're attached in the center by the centromere. That's the point that the centrioles and the spindle fibers are going to hook onto. The centrioles move to the opposite poles. Really good terminology to use there, opposite poles. The nucleolus disperses because the chromatin is now being wound around the histones to become visible chromosomes. And this stage ends with the nuclear membrane breaking down. We don't want the nucleus to be inside the cell at the moment because it'll just get in the way. So the cell breaks down the nucleus to allow the genetic material to move around to each section of the cell. The next stage is metaphase. I always remember it because the chromosomes are in the middle of the cell. What happens is the centrioles send out spindle fibers which hook onto the centromere of the chromosome. So the spindle fibers form and hook from the centrioles to the chromosomes, and they do so at the centromeres. What then happens is they will start to reel in the chromatids, but that happens in the next stage. Metaphase finishes with them lining up in the center, the spindle fibers attaching. Anaphase is the next stage of mitosis, and this is recognizable because the chromatids have been pulled to the poles of the cell. They are usually shown in transit, so they're moving towards the poles there, because what happens is they move along the spindle fibers. I always like to think of the centrioles as a bit like fishermen hooking the chromosome and reeling it in towards them. And that's pretty much what happens. We end up with the centromere splitting and the chromatids moving to the opposite poles. And it's done by these little motor proteins which walk along the track of the spindle. There are loads of mitochondria needed for this because it takes a lot of energy to move those cellular components about. Next stage is telophase, and that is recognizable as two nuclei within one cell. So the cell is starting to split apart, but the two nuclei are becoming visible. So the spindle fibers start to break down, they're no longer needed. The nuclear membrane starts to reform about each set of chromatids in each section of the cell, and the nucleolus starts to form again. You can start to see it appearing as it does so. The final stage of mitosis is cytokinesis. This is where the cells finally separate and move apart. Cytocell kinesis movement. So there's a movement of two cells apart. The cells look like they're back into interphase because they effectively are. They're just ready to fully separate. This starts though with cleavage. Cleavage is the act of cutting the cell in half, and in animal cells it's done by an actin filament that tightens around the middle and creates a furrow which then finally splits the cell in half. Whereas with plants, it sets up a cell plate in the center which forms from vesicles, and then the cell wall is made between them, cutting the cell in half in that way. The last bit I want to look at is the graphs associated with this. It is two graphs in one. You've got increase in cell mass on the left and increase in DNA in the nucleus on the right. So both lines represent a rise as the chromosomes are replicated. So you've got the cell mass increasing as the genetic material is being replicated, and also the cellular material is being replicated as well during G1. And the line on the DNA mass is the true synthesis of genetic material. You've then got the flattened period in the DNA mass there as the cell is undergoing division but hasn't fully divided. And then when it drops down, that decrease line there at approximately 24 hours is the cell actually dividing into new cells. Hope that helps. Thanks a lot. Bye.